Hi, my name is Dr. Gokul Kesavan. Today I am going to talk about genome editing while focusing on CRISPR, and the title of my talk is CRISPR, a game changing genome editor. So let's look into the details. What is a genome editing tool? It has the ability to change DNA at a very precise, predetermined location. So it's not a random location. The precision comes from the DNA sequence that we have chosen. And what is CRISPR? CRISPR stands for clustered, regularly interspaced, short palindromic repeats. So what does it mean? That means it's a short DNA sequence. It's usually 23 base pairs. These are clustered in the genome, but they are regularly interspaced and they are palindromic in nature. That means to reading from the left to right or right to left gives you the same result. So the CRISPR was originally identified as a bacterial immune system to recognize foreign DNA like viral DNA and inactivate them. But in the recent years, we know that it is such a powerful technique that it can revolutionize medical research, biotechnology, and agriculture. This is also highlighted by the Nobel Prize in Chemistry for the year 2020 for CRISPR pioneers. So let's just look into the basics of genome editing. I mentioned that CRISPR, but there are other genome editing tools like TALEN, zinc finger nuclease, and meganucleases are also can edit genome. So what do they do is like when you supply the CRISPR components, they go into the nuclei, bind to the DNA at a specific precise location based on the DNA sequence that I have chosen and cause a double-stranded break. Now the DNA is broken. So there are two ways to reattach this. One is by DNA repair mechanism. It's also called as the non-homology end joining mechanism, where random base pairs can be added or deleted, and thereby this broken DNA sequence is reattached, and this can cause a gene disruption. And the other possibility is that we can provide a repair template, which is also a piece of the DNA with homology arms on both sides. In between, we can have a transgene, for example, a green fluorescent protein gene, and by this method, we can insert a transgene at a precise location. So what are the applications of CRISPR? So as I mentioned, when the non-homology end joining happens, it is a random insertion or deletion of base pairs. This could lead to a frame shift mutation or a stop codon, thereby the protein may not be formed or the protein is prematurely terminated. So this way we can create a mutation and study gene function. Another possibility is, as I mentioned in the repair template, if we provide a transgene, we can make a transgenic animal or a plant. And the third and the most important application is that we can reverse a pre-existing mutation and correct it, and thereby we can treat genetic disorders. I'm going to explain that in detail. So there are two ways to use CRISPR in the clinical therapy. One is an in vivo method where we can provide the CRISPR components to the target tissue of interest, and the genome editing happen within the body of the patient. And other method is ex vivo method where the cell of interest can be taken out and genome edited and then infused back into the patient. Such a method was really successful and shown by a recently published paper in the New England Journal of Medicine, where they have used the CRISPR-Cas9 gene editing tool to treat two diseases, the sickle cell disease and beta thalassemia. What they have done is to take cells from the patients, and these are the hematopoietic stem cells, CD34 positive cells. They have gene manipulated these cells and infused back into the patient. And this caused a tremendous improvement in the life of the patients. This shows a promise that several other genetic disorders could be treated this way. So now I've given you a brief overview on the CRISPR, how it works. But stay tuned for future videos where we would be telling you more in detail on designing and delivering components for creating mutations, choosing target site and how to generate sgRNA, how to verify the mutations and validate phenotype, and also to create transgenic animals like fluorescent reporters and fusion proteins. Thank you very much for your attention, and bye.